So, hello viewers, this is Dr. Padala Thirupathi, Assistant Professor, Department of Botany. మనం ప్రీవియస్ లో జరిగినటువంటి ఇది ఏపీ సెట్ కి సంబంధించినటువంటి లైఫ్ సైన్సెస్ జీవశాస్త్ర విభాగానికి సంబంధించినటువంటి సెట్ ఎగ్జామినేషన్ కి సంబంధించినటువంటి ప్రీవియస్ పేపర్ ను విశ్లేషించుకుంటున్నాం. దాంట్లో భాగంగా ఈ రోజు 2012లో జరిగినటువంటి 2012లో జరిగినటువంటి ప్రీవియస్ పేపర్ ను మనం విశ్లేషించుకుంటున్నాం ఈ రోజు. ఓకేనా? సో ఈ ప్రీవియస్ పేపర్ కి సంబంధించి మనం విశ్లేషించుకునేటువంటి క్రమంలో మనం ఒక్కొక్క క్వశ్చన్ ఏ విధంగా మనకి ఫ్రేమ్ అయ్యాయి, క్వశ్చన్స్ ఏ ఏరియా నుంచి ఫ్రేమ్ అయ్యాయో మీరు గమనించాల్సినటువంటి అవసరం ఉంది. ఇది 2012లో జరిగినటువంటి పేపర్ 2 లైఫ్ సైన్సెస్ కి సంబంధించి మనం ఇప్పుడు చూస్తున్నాం. 2012 జరిగినటువంటి లైఫ్ సైన్సెస్ పేపర్ 2 కు సంబంధించి మనం చూస్తున్నాం. ఇందులో పేపర్ 2లో మనకి ఇందులో ప్రత్యేకించి మనకు 50 ప్రశ్నలు ఉన్నాయి. ఒక్కొక్క దానికి 2 మార్కులు ఉంటాయి. మొత్తం ఇది 100 మార్కులు. ఇది ఇప్పటి మోడల్ కాదండి, ఇది ప్రీవియస్ మోడల్. ఇప్పుడు మాత్రం ఆన్లైన్ లో ఉంటుంది. ఓకేనా? ఇది మన ఏపీ సెట్ అనేటువంటిది ఆన్లైన్ లేదా ఆఫ్లైన్ లో ఉండొచ్చు. మొత్తం 2 పేపర్లు ఉంటాయి, అంటే కలిసే ఉంటాయి. ఇంతకుముందు మాదిరిగా ఇది సెపరేట్ గా ఉండడం అనేటువంటిది ఉండదు, అంతా ఒకటే. అంటే లైఫ్ సైన్స్ అనేటువంటిది ఒకటే పేపర్ ఉంటుంది. అదేవిధంగా పేపర్ 1 అనేటువంటిది జనరల్ గా ఉండేటువంటి ఆప్టిట్యూడ్, టీచింగ్ ఆప్టిట్యూడ్, రీసెర్చ్ ఆప్టిట్యూడ్, అదేవిధంగా మనకు మెంటల్ ఎబిలిటీకి సంబంధించినటువంటిది పేపర్ 1. పేపర్ 2 అనేటువంటిది కన్సర్న్ సబ్జెక్ట్ లో మనకి ఎగ్జామ్ ఉంటుంది అనమాట, ఏపీ సెట్. అయితే ప్రీవియస్ లో జరిగినటువంటి ఏ విధంగా ఉంటే మొత్తం 3 పేపర్లు ఉండి, మొదటి పేపర్ జనరల్ అవేర్నెస్, అంటే మీన్స్ దాంట్లో భాగంగా క్వాంటిటీ మన ఏది టీచింగ్ ఆప్టిట్యూడ్, రీసెర్చ్ ఆప్టిట్యూడ్, దాంతోపాటు మెంటల్ ఎబిలిటీకి సంబంధించినటువంటివి కూడా ఉండేది. ఇప్పుడు అలా కాకుండా ఏం చేశారంటే పేపర్ 1 అలాగే వచ్చి పేపర్ 2 పేపర్ 3ని మొత్తం కలిపేసిన అనమాట. ఈ విషయం మర్చిపోవద్దు. అయినప్పటికీ కూడా మనము ప్రీవియస్ పేపర్ను ఒకసారి విశ్లేషించుకుంటే క్వశ్చన్స్ ఏ విధంగా ఉన్నాయి, దాంట్లో ఆ క్వశ్చన్స్ కి సంబంధించి ఆన్సర్స్ ఏ విధంగా మనం ఆప్ట్ చేయాలో చూద్దాం. సో ఫస్ట్ క్వశ్చన్ ఆఫ్ పేపర్ 2 ఆఫ్ ది లైఫ్ సైన్సెస్. సో యూకారియోటిక్ mRNA కెన్ బి సెపరేటెడ్ ఫ్రమ్ ది టోటల్ RNA బై. సో RNAను సెపరేట్ చేసేటువంటి విధానం ఇక్కడ యూకారియోటిక్ mRNA. So this eukaryotic mRNA can be separated from the total RNA by affinity chromatography. The method we can adopt, that is affinity chromatography. The second question, which of the following involves the combining of 2 cells without cell walls of different plants? So without cell wall, that cell we can call as protoplast. So without cell wall, the cell we can call as protoplast. So majority of the all animals and human beings also having this protoplast only, because of cell wall is only present plant cells. Okay, so without cell wall only protoplasts are there. So fusion of these 2 cells we can call as protoplast fusion. Okay, protoplast fusion. The inducing of this protoplast fusion by PEG, polyethylene glycol, PEG, which is induced the protoplast fusion. So this we can call as fusogen. So PEG also we can call as fusogen, which is PEG, polyethylene glycol. Okay, then the third question is non-polar amino acid residues are found mostly. So these generally the amino acids, they are polar amino acids and non-polar amino acids, which we are observed in the different parts of the plasma membrane, especially in the cell. So these non-polar amino acid residues are found mostly in the core of proteins, Then the fourth one is, which of the following organelle does not carry out transcription? Remember, my dear friends, actually transcription takes place in only nucleus. Nucleus is only responsible for the transcription. Okay, so transcription means from DNA to the formation of the mRNA or production of the mRNA from RNA, the synthesis of mRNA from RNA. So that we can call as transcription. Transcription takes place in nucleus only. But along this nucleus, some of the organelles responsible for this, along with nucleus, the transcription is also takes place. Some of the organelles like chloroplast and mitochondria. Why? Because this chloroplast DNA or mitochondrial DNA, cpDNA or ctDNA or mtDNA, which is a circular DNA, which are present in the stroma of the chloroplast and the matrix of the mitochondria. So that's why the mtDNA and ctDNA which are present in these cell organelles. So that's why these are all shows about the transcription. Okay. So along with nucleus, chloroplast and mitochondria takes place transcription. But peroxisome is not responsible for the transcription, which is responsible for the photorespiration. Okay. Peroxisomes are responsible for the photorespiration. Okay. Then the fifth one, so ascertain and region. So these type of statements which we are observed in paper 2 and paper 3 of the life sciences are APs, any set of question paper. So ascertain, A, hexokinase and glucokinase have low and high Km towards glucose respectively. And the reason is glucokinase operates under the high glucose concentration only. So among these, between these two statements, which are correct statements. So both A and R are correct. Both A and R are correct. So A means assertion. B is reason. So both statements are correct regarding of this glucose or glucokinase or hexokinase activities. Then the sixth one, also assertion and region. So in this, proto-oncogenes are found in most of the animal cells. Okay, proto-oncogenes. Oncogenes means the genes are responsible for cancer. Okay, they are related to viral oncogenes. So between these two statements, which statements are correct? So both A and R are correct. So proto-oncogenes are found in the most of the animal cells and they are related to viral oncogenes. Okay. Then the next one, the seventh one is also A and R, assertion and region. All eukaryotic mRNAs are capped at 5' end, and 5' end cap is not essential for efficient mRNA translation. So between two statements, which are correct? So between two statements, A is correct, assertion is correct, but R is wrong. Okay. All eukaryotic mRNAs are capped at 5' end. And the eighth one here, the match the following: nuclear magnetic resonance, radio waves; electron spin resonance, microwaves; Raman spectroscopy, infrared waves; spectrophotometry, ultraviolet or visible light. Okay, spectrophotometry related to ultraviolet or visible rays or visible light. Visible rays. This is related to spectrophotometry. So Raman spectrometry related to infrared, IR rays. Okay, and the electron spin resonance, microwaves, and nuclear magnetic resonance, that is radio waves. Then another matching this BT cotton related to pest resistance varieties. Okay, Bacillus thuringiensis mediated cotton. It is a transgenic plant. So in this question we are observed, these are all transgenic plants. So among these plants which are suitable for the correct option. So BT cotton is responsible for pest resistance, and flavor saver tomato. So tomato, it is improved the shelf life. So which is very fresh manner, so many days, which is preserved. And the Roundup Ready soya sorghum, which is related to herbicide tolerant. And golden yellow rice, that we can also call as golden rice. So golden rice, which is highly nutritive quality, that is vitamin A. So beta carotene or vitamin A mediated rice, that is golden rice. Okay, Taiwan Nature variety is involved in this preparation of the vitamin A contained rice, that is golden rice. The 10th one, virus that persist in the cell and cause recurrent disease are called, that we can call as latent viruses. So we are observed in viruses, the lytic life cycle and lysogenic life cycle. So in that cycles, we are observed latent phases or latent viruses. Okay, they are persistent in the cell and cause recurrent diseases. Then the 11th one is A and R, assertion and reason. So in this, indirect somatic embryogenesis, embryos are developed without going through callus formation. That is, we can also call as indirect somatic embryogenesis. And this is possible due to the presence of pre-embryonically determined cells. In this, both A and R are true, and R is correct explanation or reason for A. R is correct explanation of the A. That means indirect somatic embryogenesis or embryos are developed without going through the callus formation, without formation of the callus, directly these embryos are formed. Then this is possible due to the presence of pre-embryonically determined cells. Yes, both are correct. At the same time, R is correct explanation for A. And 12th question, in complement cascade, the complement 1 activates as follows. So in these four C1Q, C1R, C1S, C1D. These are the sequences for the complement cascade. C1Q, C1R, C1S, and C1D. And the 13th question, this is also match the following. Pyridoxal phosphate. So pyridoxine, that means vitamin B6. So in this, we are observing the cofactors are especially coenzymes are some of the vitamins we are observed in these questions. Pyridoxal phosphate, nothing but vitamin B6, and niacin, that is vitamin B3, thiamine pyrophosphate, that is vitamin B1, and flavin nucleotide, that is vitamin B2. Okay, means riboflavin or flavin nucleotide. Okay, then the 14th one, the protein structure is stabilized by. Among these four options, 1, 2, and 4 are the correct statements. That means the protein structure is stabilized by hydrogen bonds, and also along with hydrogen bonds, dicyclic bonds also involved, and hydrophobic interactions caused for the protein structure stability. The stability of the protein structure caused by hydrogen bonds, disulfide bonds, along with hydrophobic interactions. Okay. Then the 15th one, in this two-dimensional electrophoresis, okay, electrophoresis involves IEF in first dimension and SDA space in second dimension. Okay, first dimension, that is IEF, and second dimension, that is SDS page. Okay, first dimension is IEF, second dimension is SDS page. That is two-dimension electrophoresis involved in the first dimension IEF and second dimension SDS page. And the 16th one, electron transportation chain, ETS, contains the following components. My dear friends, remember the electron transportation chain which we are observed in the mitochondria, especially in the inner membrane, that we can call that as a cristae. So cristae shows about the electron transportation chain or electron transport system. This system contains 5 complexes. Among these 5 complexes, 4 complexes we are observed here. This is the sequence. My dear friends, remember, this is the sequence. The first complex contains this component, that is NADH, nicotinamide adenine dinucleotide hydrase, that is NADH, CoQ oxidoreductase, that is first complex involved, this component. And second one is succinate CoQ oxidoreductase. And the third one is coenzyme Q, that is the cytochrome oxidoreductase component. And the fourth one is cytochrome C oxidase. So these are the 4 complexes we are observed in electron transportation chain. These are the important components, these are the sequence. So first one is NADH, CoQ oxidoreductase. The second one is succinate oxidoreductase. The third one is coenzyme Q, cytochrome oxidase. And fourth one is cytochrome C. The third one is coenzyme Q, cytochrome oxidoreductase, and the fourth one, last one, that is cytochrome C, oxidase. Oxidase is last one, reductase, oxidoreductase is third one. Okay, then the 17th question, correct order of the chromatin organization in chromosome. Remember, my dear friends, generally chromatin we are observed during the cell division, or even though the chromosome structure, we are observed in the nucleus, that is see here, and also chromosome. The sequence, the order of the sequence, the chromatin organization order is, correct order is, first one double-stranded DNA, and the second one is the nucleosome, and third one is the solenoid, and fourth one is the chromosome. So chromosome structure we are observed here. This is the chromosome structure. In this chromosome structure, we are observed, that is this contain chromatin thread, that is nothing but a DNA, double-stranded DNA, dsDNA, and the dsDNA along with the beaded-like structures we are observed in the nucleosomes. Okay, that we can call as histone octamers, histone proteins, histone octamers. 4 pairs of the histones are involved in the nucleosome, and also they are going to coiling. They are going to coil, that we can also call as solenoid, and the last one is the finally chromosome is formed. Okay, this is the chromosome. And the next 18th question, give the correct sequence of the following steps and use southern hybridization. Remember, my dear friends, the southern blotting related to detection of the DNA, northern blotting related to detection of the RNA, or western blotting related to detecting of the protein. Here, the southern hybridization contain different steps. The steps, the sequential order of the steps are: number one, restriction, digestion, and the second step is separation by electrophoresis, that is gel electrophoresis, and the third one is blotting membrane, and the last one is detection. Okay, this is southern hybridization. This related to detecting of the DNA fragments. Okay, and the 19th, according to ICBN rules, we know that International Code of Botanical Nomenclature, the suffix of the family should be ends with AC, whereas prefix refers to. So prefix refers to, generally, my dear friends, the AC, Solanaceae, Liliaceae, Asteraceae, okay, Convolvulaceae. So this is the suffix. The name of the suffix should be ends with AC. But whereas the prefix refers to, remember, my dear friends, that is always generic epithet. That is generic epithet. Okay. Then the 20th question, pick up the correct combination of the following using valid name and part used. See here. In this, among these 4 options, this is the right option, that is millets, means sorghum vulgare, belongs to Poaceae family. Okay, and the fruit we can call as Caryopsis, and pulses, the scientific name of the pulses, especially Kajanas kajan, belongs to Fabaceae family. So we are observed cotyledons. Okay, in seed we are observed cotyledon. Caryopsis means there is no differentiation between. Okay, there is no differentiation between fruit carp, and even though the what we can say that the seed coat, there is no differentiation. So that's what that we can call as a Caryopsis. So pulses, cotyledons, and fibers, generally Malvaceae family. We are observed fibers, Gossypium herbaceum, that we can also call as cotton, belongs to Malvaceae family. Especially epidermal hairs of the seed, we are used for these fibers. Pulses comes from cotyledons. So the millets comes from Caryopsis. Okay, even though our rice means rice, the paddy belongs to, that is what we can say that Oryza sativa, that belongs to Poaceae or Gramineae family, Poaceae family. The fruit we can call as Caryopsis. And the last one is the fruits. The fruit plant, that is Mangifera indica, belongs to Anacardiaceae family. We are used that contains epicarp and mesocarp. We are using epicarp and mesocarp. Epicarp and mesocarp. Both the pots we are edible pots from the drupe. Okay, so drupes, the best examples of the drupes are coconut, and what we can say that the Anacardium occidentalis, Anacardiaceae family, Cocos nucifera, that belongs to Palme or Araceae, and here the Mangifera belongs to the Anacardiaceae family. These fruits are drupe. Okay, drupe type. And the edible part of this fruit is epicarp and mesocarp. Endocarp is highly, what we can say that the sclerenchymatous cells we are observed in the endocarp. Okay, then the last, the 21 question, that is assertion and region, A and R. Apospory. So apospory is not uncommon in some ferns like terrace. Apospory. Okay, apospory. In ferns, sometimes the prothallus is formed from vegetative parts of the sporophytic plant. Here, my dear friends, both statements are true, and R is correct explanation to A. Okay, correct explanation to the A. That means apospory. Apospory is not uncommon, means it is a common in some ferns like terrace plants, and in ferns, sometimes the prothallus is formed from vegetative parts of the sporophytic plants. So that's why the formation of the prothallus from the vegetative parts of the sporophytic plant we can call as apospory. And the 22nd question, the Biosphere Reserves is recently notified by the government of India, that is Seshachalam. Here, the Biosphere Reserves in India, especially, we are absorbed. That means 26 Biosphere Reserves we are absorbed in the entire. The government has identified Seshachalam Biosphere in Andhra Pradesh, that is Seshachalam Hills. Okay. Then epiphytes are rare in Delhi because it is semi-arid climate. My dear friends, remember epiphytes should be present in the epiphytic plants like Vanda and other Orchidaceae family members, which they are present in the different trees, on the bark of the trees, but the environment should be in moist climate. Okay. So that's why the Delhi is related to semi-arid climates. That's why epiphytes are very rare. And the 24th question, arrange the progressive manner. Arrange the progressive manner of the ecological succession in the zeros here. So zeros here related succession. In the succession, first one is rhizocarpon, and the second one is funaria, and third one is the poa, and last one is tree. These are the sequential of the zeros here. Okay. And 25th question, match the following: aquatic habitat, that plants we can call as hydrophytes, community abundant light we can call as heliophytes, and community in shade, that is zeophytes, and last one is the dry condition that we can call as especially xerophytes. Okay. And the 26th question, pick up the source from the air pollution from the following, that is the volcanic eruptions and dust storms and CFMS, chlorofluoromethanes. So these are all source of the air pollution. Okay. And which of the following has more redox potential? Okay, redox potential value, that is nicotinamide adenine dinucleotide, that we can call as NAD. Okay, NAD in electron transportation chain or in mitochondrial inner membrane cristae, which we are observed. These are the components or complexes that we can call as a cytochrome B oxidase, cytochrome B, cytochrome C oxidoreductase, ubiquinone, and nicotinamide adenine dinucleotide. So that is NADH. See here, it is the high redox value, that is nicotinamide adenine dinucleotide, and the RQ value of the simple sugar should be equal to 1. Okay, RQ, respiratory quotient value, always 1, equal to 1, that is glucose or sugar. Okay, carbohydrates, RQ value always 1. Then protein value, that is 0.9. And the Fats RQ value 0.7. More than one organic acids. Okay, more than one organic acids. Infinitive, especially infinitive. That is, we can call as especially succulent plants. Okay, then the assertion and reason below statements are here. The pronounced decrease in quantum yield to at red zone, that is greater than 680 nanometers in photosynthesis, is termed as red dot. This term has given by Robert Emerson. Robert Emerson has given this red drop term. So the discovery of the Emerson effect has clearly shown the existence of two photosystems. Yes. So two photosystems are 100% just because of the discovery of the Robert Emerson's effect, that we can call as Emerson enhancement effect. Emerson enhancement effect is also observed red drop, and this is caused by the identification or the existence of the two photosystems, that is photosystem 1 and photosystem 2. So both are correct. And arrange the following components order in which they appear in Krebs cycle. So Krebs cycle, we are observed the products or the intermediates of the Krebs cycle. So hence Krebs has clearly stated 10 stages of the Krebs cycle, that is citric acid cycle, also tricarboxylic acid cycle. In this manner, succinic acid, fumaric acid, malic acid, then oxalic acid. So this is the order. Okay, then 31 question. Which one of the following combination chromosome number N and DNA content, that is C? So 1N and 2C is the right answer of the mammalian oocyte. That mammalian oocyte, the stage is diplotin stage. Diplotin stage of the mammalian oocyte, the number of the chromosome is 1N. The chromosome number that is means DNA, that is 2C. Okay, then assertion and reason 32 question. See here. As polyspermy is being slowly blocked, the cortical granules explode and release their contents into the previtelline space. The quick blocking of polyspermy sets in depolarization of the egg plasma lemma and mobilization of the calcium ions from stores within the egg. So here both A and R is true, but R is not correct explanation to the A. And 33 question pairs the match the following: telencephalon hippocampus, mesoencephalon midbrain, and diencephalon optic vesicle, and the myeloencephalon cerebellum. Okay. And 34th question, during neural tube formation, the chick embryo, the neural plate involves following event sequences, that is shaping, folding, elevation, convergence, and clockwise. Very, very important. Okay. Each AP set contain this type of questions. Okay, the order of evolutionary pattern of nitrogen excretion in animals. So the concentration of the ammonia, that is ammonia is lower concentration compared to urea. Urea is highly concentration compared to ammonia, and uric acid is highly nitrogenous component or nitrogen excretion. Okay, uric acid. And this is the formula, especially dental formula in mammalian manaki heterodontic. So 0.000033 by 3133, that is 32 teeth we are observed in sheep. Okay, and 2033 by 1023, that is 28 teeth we are observed in rabbit, and 1023 by 1013, that is 22 teeth we are observed in squirrel, and 2123 by 2123, that is 32, we are human beings. Then 37 question, assertion: absorption of the products of carbohydrates and protein digestion are against a concentration gradient, whereas the absorption of the products of lipids are different. The reason is the end products of carbohydrates, proteins are absorbed into the epithelial cells of intestine is by active transport, whereas the product of lipids are by diffusion. In this, both A and R are correct. Okay, then 38th question. The Bohr effect represents in interaction between. So my dear friends, remember the Bohr effect is nothing but O2 equilibrium and proton dissociation of the hemoglobin. Okay, O2 equilibrium and proton dissociation of hemoglobin. The 39th question, see here combination expected, chromosome combinations, they expected then offsprings of the obtained from selfing of the plant having capital A, capital A, capital B, capital B, capital D, capital C, capital D, capital D. So homologous pairs. See here, my dear friends, the correct sequence is AA, BB, CC, DD because of homologous pairs are here. Then the 40th question, match the following. ABO, that allele is 5. MNSC, allele is 20 plus. P, blood group, that is allele is 4. And rho factor, RH factor, 30 plus alleles we are observed. Then assertion, gene is a biologically entity, a heritable function detected by observing the effect of mutation. A gene consists of all the DNA sequences necessary to produce a single peptide or RNA product. Then R is, the gene is a single contiguous a protein encoding stretch of DNA. In this context of the above statement, which is the correct? My dear friends, both A and R are correct, but R is not correct explanation of the A. Then the 42 question, sex-linked genes on the entire X chromosome in Drosophila melanogaster mapped, found we can call as 68 map units. Drosophila melanogaster, 68 map units. And the 43, what are the two theories that are combined in the synthetic theory of evolution? That is, we can call as Darwin theory of natural selection and Mendelian theory of heredity. Okay, Mendelian theory of inheritance. See here, two theories that are combined the synthetically theory of evolution. Synthetic theory of evolution combined by Darwin's theory of natural selection and Mendelian theory of the heredity. And the 44 question, which of the various types of the isolating mechanisms of the prevent gene exchange between population related species? See here, habitat isolation. Habitat isolation related to population lives in same. And seasonal isolation. Seasonal isolation related to regions but occupy different habitats, that we can call as seasonal isolation. And ethological isolation. Ethological isolation that represents, that is, populations are isolated by different and incompatible behavior. And last one, mechanical isolation, that is restricted by differences in structure of reproductive organs. Okay, then 45th question. Arrange the following is in a row leading from more like reptiles or more like mammals. See here. The following fossils are, number 1, Polycasuria, Pelicosaria, and Therapsida, and Cynodonta, and Mammalia. These are the order. And 46, the finch's beaks evolved to be larger in times of food shortage and the smaller in times of abundance. So we are observed, Charles Darwin observed in the Galapagos Islands, the finch beaks or finch bird beaks are evolved and the food supply fluctuated through times according to the weather. Here, my dear friends, so both are correct and the R is correct explanation to the assertion. Okay. Then the 47 to 58th question, paragraph is here. The 47 question, helical winding DNA is removed by an enzyme. My dear friends, removed by topoisomerase. Okay, topoisomerase. Then 48th question, discontinuously synthesized lagging strand DNA fragments are sealed by which of the enzyme? That is DNA ligase. DNA ligase we can also call as molecular switches. And 49th question. So In which one of the following four statements, which one is the incorrect statement? See here, this is the incorrect statement. Remaining are all correct statements. Leading strand by synthesized in 5' to 3' direction. It is also correct. Lagging strand is synthesized by back stitching process, right? It is also correct. And the last one is the anti-parallel strands of DNA are separated by helicase, right? So the incorrect answer is DNA polymerase can directly synthesize new chain of DNA. This is the wrong answer. Incorrect. The last question, which one of the following four statement is correct? So leading and lagging strands are synthesized in a different manner because DNA strands are anti-parallel. So this is the last question of the AP set 2022 question paper. Okay, so remaining AP set 2012 paper 3 will discuss next class. Okay.